Hi, thanks to the organizers for letting me be here. The original title was Alternative Biopolymers in Early Evolution, but I figured I should be a little more specific about what it is that I want to present today. And so I chose this subtitle, Assessing the Evolutionary Potential of Nucleic Acid Libraries with Increased Information Density. And what I would like to show you today is a set of experiments that we started in the attempt to understand what is the real information density that is needed for a library to achieve evolution while still maintaining the least possible amount of, while still using the least possible amount of energy to do so. So why would we want alternative biopolymers to achieve that? One of the reasons is, of course, that RNA probiotic chemistry is hard and only works under very specific sets of conditions, and we heard about it all through this conference. Actually, this morning, Tyler Roche made a few good points about that. Also, this one, I should have erased it after this past talk. This was very good, actually. So I think that replicases still are a work in progress, but people, as we just saw in the Rao lab and in Holliger's lab and in Joyce's lab, actually they're doing great things with them. But yet, replicases are a key player in a hypothetical RNA world and are still a little bit of a challenge that needs to be overcome. Another point is that alien life arising independently of life on Earth might have had a different molecular biology than what we see or was experienced here in the past. And this is also something that people have been talking about quite a bit these past few days. But even if the biopolymer of choice had been the same, alien life might have chosen a different evolutionary pathway, no matter what. And even without having to go to other planets or before or after the evolution of life, today's natural RNA post-transcriptional modifications are seen by many as remnants of an RNA-like world, and they might be considered as hints that in prebiotic times, these modifications could have arisen during rather than after the establishment of the biopolymer. So in other words, these features that we call modifications or alternative now and here might have been the main evolutionary players in other times and planets. I did like this, to add this quote from yesterday morning's talk from Christine Keating that I think summarized well what a lot of people have been saying these days. What happens, so there's this dichotomy in which what happened versus what could happen or could have happened. In this dichotomy, the second is a much larger group of reactions, so we need to start to investigate this. I'll go back for a second. So what, as I said, what we decided to do is to set up a series of experiments, a couple of initial in vitro selections, starting with libraries. So the two selections that we want to do in parallel would go towards the same function, but starting with two libraries that are the same length, but carry different information contents. So how do we achieve this enrichment or depletion of information? Nothing would make me more happy than being able to do it with Nick Hutt's systems or the nucleosides that Taylor showed this morning, but we are still lacking a molecular biology for those systems. So what we do have in the lab, actually at home at FAME, is this artificially expanded genetic information system that was developed in Benner's lab in the last 20 years. And for this system, we do have a very well-established molecular biology, structural biology, synthetic biology, and we have shown in the past years that we can actually use this system for evolutionary experiments easily and effectively. So the idea behind AEGIS is to complete the Watson-Crick pairing concept, as it's shown in this scheme, by shuffling hydrogen bond donors and acceptor groups, forming additional orthogonal nucleobase pairs. They resemble natural nucleotides in size, shape, and pairing geometries. They are independently replicable, do not interfere with the RNA folding or the DNA double helix structures. They increase the information density, and they have the potential to increase functionality, and that's what we want to test. So what we have done in these preliminary studies is we started out by using these pairs, Z's and P's, where Z has an extra nitrogen group on N5, while and also has an extra hydrogen here at N3, and P is lacking hydrogen right here and has an extra nitrogen in N5 too. So we picked this couple at first because it's the one that we have studied more and we know it works, and it's a first step toward increasing the diversity and the information inside the library. Now, the next step is to try to understand what is the sequence space coverage that we are trying to achieve. As I said, we are going to start with same length libraries, but one will have 6 letters DNA and the other one will have 4 letters DNA. If you look at this table, well, sequence space is defined as, for us at least, is defined as the number of library bindle blocks to the n, where n is the length of the library. So if we have 19 nucleotides and 6-letter DNA, we will achieve 100% coverage when we use 1 nanomole of material, which is what you usually are able to handle in a laboratory experiment. While if we have 4 letters DNA for the same amount, you will actually have about 1,000 molecules, 1,000 copies for each sequence that you have in your library. So obviously a 4-letter DNA always has an advantage compared to a 6-letter DNA when you're trying to do wet lab experiments, evolutionary experiments at least. So if you look at the table and you keep going through the numbers, you will actually see, I'll do the math for you, you will actually see that for every, there is the space coverage decreases of 1 order of magnitude every 5 nucleotide length increase in the library for 2 bits of information difference. So the longer you go, the more you actually have the 4 letters DNA have an advantage versus the 6-letter DNA. That is, unless the functionality and the extra nucleotides that you're adding are actually more powerful, more effective for the type of phenomenon that you're trying to evolve. Down here we actually have the actual libraries that we have used for these preliminary studies. We started with an N25. For the 4 nucleotides, we started with 0.5 nanomoles. This was a first run. So we covered 25% of sequence space for the standard DNA library, while for the AEGIS library, we actually covered 0.0011% of the sequence space. So we did, the next step would be which reaction are we going to pick. As a proof of principle, probably one of the easiest reactions to pick is selecting for RNA-cleaving DNazymes. Raghav talked about it a little bit this morning. It's a very well-known type of molecule. It's very well studied, so we have a lot of literature to compare to once we get some results. And people have actually been done selection on DNazymes for the past maybe decade. And they were, of course, first selected by in the Joyce lab, Santoro and Joyce, 1997. This is Craig. Craig Jerome did all of it. He's a technician at FAME. He's a very skilled guy, but he needs still a little training. This is the schematic of the selection. So we start with an RNA target that is here in blue, that is attached to a primer that will amplify a library, and at the 5' end actually has a biotin, so that you see after you get the double-stranded DNA, you can remove one of the strands, and everything, all your system will be attached to the biotin. This portion, which is your evolving ribozyme, can fold over the target, cleave it, and you will actually have in the supernatant your evolving library that you can amplify. And this is just slightly modified from the original protocol to make it a little more effective and remove a few steps. These are the conditions that we use. This 2 millimolar magnesium, because everybody cares about magnesium. And what we care about is that we use pH 7.8 for a very specific reason that I will say in a moment. And And okay, so this is the big deal. We did the parallel in vitro selection, and what we could see is that while the standard DNA had a really hard time growing, at least in our conditions, at 25 nucleotide long library and with only 2 millimolar magnesium, and instead the AEGIS, the non-standard library grew very well and was happy. So this by itself is already a surprising result, because while we did expect that AEGIS might have some advantages because of the extra groups that we had, we didn't expect that we would have such a big difference. Also, I'm a little disappointed. We did this 2 or 3 times, I think 3 times in total. We really can't grow these DNazymes with standard nucleotides. It really looks like only 4 is not enough for 2 millimolar magnesium. Thank you. And for 25 nucleotides long sequence. One reason might be in the Z nucleotide, which I reported here again, which has a pKa of 7.8, which is exactly the pH that we use during selection. So ideally, you can see here the output of the deep sequencing that we did. So we actually were able to, we have a technique to transform our AEGIS DNA into standard DNA, to submit it to deep sequencing, and then keep track of where the original Z's and P's were. So we applied that technique to these libraries, and we actually sequenced each cycle of the selection. And what was surprising, but maybe not, is that there was a preponderance of Z's in all the major clusters that survived the selection. Here, there's just a graphic representation of how the various different clusters appear and disappear during the selection. This is a table that shows you at which cycle each cluster will appear. But back to the Z's, so our main sequence, the cluster that actually showed most prominently in the deep sequencing analysis, actually carries 4 Z's, and it has 2 sites of cleavage. So in the hypothesis that the pKa of Z is actually very important, here you can see how half of the molecules will be protonated, half of the molecules will be deprotonated, and they will act each of them, every couple, around one of the sites of cleavage to achieve cleavage. So this is one example of all the cheese-acting clusters that we found. We got 3 families of AEGIS DNazymes. One, as I just said, is cuts at U9 and U13, C1, 2, 6, and 7, cluster 1, 2, 6, and 7. Then we have cleavage at A16, and then we have cleavage at A10, sorry. So we got these 3 families, and we decided to go ahead and try to characterize exactly what was happening in each of these clusters. We took cluster 1 to begin with. So in cis, it will just have a decent activity, but this is a really big molecule. You do see that the evolving portion of the library actually does best pairs very nicely with the RNA. And then you have the Z's over here. So what I think is happening is actually that we're getting a very big 3D restructuring around the partially double-stranded area. This is actually, there's a GU wobble, and here it's probably open because there's this huge unstructured area. But we checked that C1 with no Z would not actually have the same activity, and that's what is shown here in 2 really different representations. So here you're monitoring the appearance of the bands that will show you cleavage. Here you're monitoring the disappearance of the full-length product. And the red one is the one that does not contain Z, and the black one is the one that does contain Z. I'm running out of time, so I'm just going to go very fast. We did find, this is it's a very dense slide that basically we did a bunch of biochemistry assays in which we separated the 2 strands, and we finally could see that we get a Kcat of 10 to the 9, we get a Km of 10 to the 5, and a catalytic efficiency of about 10 to the 5th per mole per minute. That is very, very comparable to, I don't know if you guys know the work of David Perrin. He works with heavily modified DNazymes, and he got exactly the same type. Well, we got similar catalytic efficiencies just with 1 extra nucleotide. So that's very interesting. conclusions and perspectives, this is just a summary. What we would like to do after is actually start to perform multiple parallel strand AEGIS, standard AEGIS selections with increased and decreased lengths, number of nucleotides, and extra nucleobase modifications. So 2, 4, 6, 7, 8, and whatnot. I am going to thank, of course, Craig Jerome, who is the one who did all of these, all our funding agencies, although this specific project is not yet funded, and everybody at FAME and Firebird. Thank you. I'll take any questions. If there's time for questions.
Got time for one question?
Okay.
All right. We're going to go to the back.
Yes.
So, but then M2 of guanine is critical for generate tetraloop stability.
Can you repeat that? I'm sorry.
You said the M2 of guanine hydrogen bond donor is critical for tetraloop stability.
Well, see, in other studies, actually, we did found that when these MPs and other pairs in the AEGIS system are not best paired, they actually contribute into secondary and tertiary structures that we are still investigating but seem to be extremely interesting and not seen before in DNA and RNA. What I meant with that point was that we have also a lot of data that shows that when you substitute Z and P in like double-stranded structures, you actually do get the same foldings that you expect from known molecules. You might have seen, the Hachimoto DNA paper with the spinach aptamer and some ribose switches that we actually substituted Z's and P's for. But yes, it's true. Some of these nucleotides will actually provide, I think, we don't have confirmation yet, but I really think we'll actually provide new structures for DNA and RNA molecules. Yeah.
All right, great. Let's thank Elisa again.
Thank you. Should I give this back or do I leave it here?